Thank you for inviting me to speak about our late-breaking abstract that we will be presenting at the American Society of Hematology 2020 conference. As a hematologist, some of the most common questions that we get asked by patients that have a blood cancer are, how long have I had it for? Oh, how fast did the cancer grow? And we set out to answer this in the myeloproliferative neoplasms, which are a chronic hematological malignancy. We took 10 patients with MPN who presented with disease between the ages of 20 and in their 70s. We took single blood cells from these individual patients and grew each cell into a colony of cells, and then we performed whole genome sequencing of each individual colony. In total, we sequenced up to about 1,000 whole genomes across the 10 patients. And by sequencing at such a large scale, we were then able to use all the naturally occurring spontaneous mutations that we accumulate in our hematopoietic stem cells with age. And we could use those mutations, a total of about 500,000 mutations, to draw a family tree of the relationship between the blood cells within individual patients. And by drawing those family trees, which reveal the ancestry of those blood cells, we could then time the driver mutations that cause MPN. And we know that the majority of MPNs are caused by driver mutations in one of a few genes, in particular the JAK2 gene, but many patients have mutations in additional cancer-associated genes, such as mutations in DNMT3A. What we found was quite striking. We found that the JAK2 mutation was often acquired very early in life, in childhood and possibly even in utero. Indeed, when patients where the JAK2 mutation was acquired as the first event or the only event driving their MPN, it was acquired at the very latest, up to 4 months or up to the age of about 11 years in many of our patients. JAK2 mutations could also be acquired second, though, in these diseases. In many of our patients, some of their other cancer-associated mutations were also acquired incredibly early. In 2 patients, we found DNMT3A mutations that occurred within weeks of life. So in utero, up to a couple of weeks post-conception. And DNMT3A mutations are one of the commonest mutations associated in clonal hematopoiesis. And we've often thought that aging in some way promotes the outgrowth of these mutations. But actually, what we found is it's not that aging per se drives clonal hematopoiesis, it's that these mutations simply take an age to grow. So overall, we found in these patients that these driving mutations occur very early. They grow and evolve over a lifetime to cause disease, regardless of whether disease presents in your 20s or in your 70s. Your mutations occur very early and evolve very slowly. What was also important is that the rate of growth of the clones is different in different patients. And we found that the faster that the clones are growing, the more likely you are to present with disease sooner. So what we saw is that the latency between JAK2 mutation acquisition and MPNs was over 30 years, in one patient over 50 years, and that the stronger the selection on those driver mutations, the quicker one gets their diagnosis. So this really now raises questions as to how we define MPNs, given that they have been with the patient, these driver mutations and clonal outgrowth has been a lifelong process. And it also raises questions as to how perhaps we could use this information to better early detect and prevent MPNs, because we now could use this information to both detect patients early and estimate which patients are on a trajectory to future complications. Our data is not yet published, but we hope to publish it soon. So for now, these data are preliminary, but we're very excited about the results. And the next questions will then be also about other blood cancers in terms of the trajectory of those. Which blood cancers have a shorter trajectory? Which other blood cancers or forms of clonal hematopoiesis have a lifelong trajectory? And it's really those questions of understanding the origins and the evolutions of these blood cancers that are critical for programs aimed at early detection and prevention.